Hey guys, welcome to topic 3 in chapter 15. In this topic we're going to explore those genotypic methods we talked about in topic 1. Now you haven't done these a lot in lab or at all in lab, but you may have talked about them. So it's important that you really focus on this chapter so you understand that. If you have any questions, I know this can get a little weird for those of you who don't have any kind of molecular background. So just let me know if you have any questions. Genotypic methods are really important for you to understand because this can give us the exact causing agent of disease or tell us how similar it is to something else. Another reason why we use genotypic methods is that if we don't want to grow up vegetative cells because they're a dangerous pathogen, genotypic testing will give us an identification without doing all that phenotypic work we talked about in the last topic. Here are our topics for this, or here are our objectives for this topic. As always, if you have any questions, please let me know. So one of the first tests we talk about, and I'll put videos up for all these tests in Blackboard, are genetic probes. What we do with this is we know we have a for sure sequence of bacteria. So if we're talking E. coli, we have a for sure sequence that's only found in E. coli. What we can do is we can build a probe, which is a complementary sequence of DNA with a little color or fluorescent indicator on the end. We then place this probe in the presence of extracted bacteria or extracted DNA, and if it binds to it, that gives us a positive result for the bacteria we're looking at, in this example, E. coli. So you can see here that the purple dot would indicate that's positive for E. coli. So that's how this works. It uses complementary binding, C's to G's, A's to T's, to give us that hybridization. Another one we'll do is nucleic acid sequencing or RNA analysis. Now, while probing provides us an identification, sequencing can do two things for us. If we sequence the 16S RNA, we can see how similar microbes are to each other. So say we have this new bacteria, we don't know what it is, we can sequence it and say, oh, it's really close to Staph aureus, or oh, it's this. And so it helps us to identify where it fits a little bit and then use that information to help us know how we potentially could treat this novel disease. We can also use something that combines that hybridization we talked about with a fluorescent tag and the sequencing method to do what we know as FISH. And as you can see here, what we do is we take the positive blood culture, we take the gram-positive cocci from there, and we take a probe and we place it near it, and it'll turn the cells green. This allows us to do a really quick analysis, whereas the sequencing takes quite a while. So it's important to understand how that works. But mostly with both these, it's a big thing about probing. We have complementary sequences that we do. And the big thing is, as long as we have the right sequence, this is very, very specific. The last assay, and I'm not going to go through how this works because we've already talked about this in chapter 8, is PCR. The point of why I'm talking about PCR now is that PCR is used in the laboratory when we need rapid results. Somebody is really, really sick and we need to get a result real quick. PCR, as well as all these other genetic tests or genotypic tests, are really expensive and time consuming. Whereas PCR can happen pretty quickly, but it's still fairly expensive and really sensitive. But it allows us to test a result within hours. And so this allows us to know before a 48-hour window what potentially is causing problems. So this is the end of this really short topic. As I said before, please review those objectives and let me know if you have any questions about any of these things. I know I went through them fast, and I know that they're probably the newest assays out of all the groups we're going to talk about today, or in this chapter. So please just let me know if you have any questions.